All right. So, even though I don't really have anybody joining me back, I thought that I might just talk a little bit about some antimicrobials. So I mentioned before that you guys should probably be on track to memorizing those by now. It's really a lot, and I recommend doing a little bit every day. It'll help a lot. So I wanted to talk through the idea of selective toxicity. So that's the whole idea that we're targeting the microbe, not our own cells. If we target something that the cell that the bacterial cells have that we don't have, that means that it's going to be selectively toxic towards the bacteria. So that also means that it's not going to kill off our cells, which is very important, right? Because we don't want to lower our immune system and kill our own cells while killing off the bacteria, because it's going to make us feel a lot worse. It's going to make us feel like the drugs aren't working, right? And there's not really a clinical way to tell the difference unless you keep doing cultures. So there's this idea of the toxic dosage level and the therapeutic dosage level. So we all know, like I think it's if you eat like 8 pounds of apple seeds, there's enough cyanide in those to kill you. That would be a toxic dosage level. Okay. So it's the max amount you can have before damage starts, right? But therapeutic dosage level is how much works, right? Because if we give too little of an antimicrobial, it's not going to give a hard enough punch, right? If we give penicillin and it's half of the dosage that we need it to do, it's not going to be effective against the bacteria. But if we give it at a therapeutic level, that means that it is going to be effective, that it's going to knock out that bacteria, and it's going to the patient should clear the infection. So in order to find the chemotherapeutic index, you take the max tolerable dosage, so the max you can give before harm, and the minimum therapeutic dosage. Okay. So if we have a drug that like if you have, you know, let's say for the first example that it is those like 8 pounds of apple seeds that have enough cyanide to start hurting you, right? So that would be our max our max tolerable dosage would be like 8 pounds, right? But we usually have, and what does the trick is like the 4 or 8 seeds that come in a normal apple, right? Those don't hurt us and they do the trick. They're in a normal serving, right? So our therapeutic bottom number is going to be, let's say for the sake of ease, 8. But we have a toxic dosage level of 8 pounds. So that could be, I'm going to say 800. So if we take 800 and divide it by 8, we're left with 100, right? So the chemotherapeutic index would be 100 for that one. Now, let's say that there's a drug, vancomycin, for example. It doesn't have the highest, or it doesn't have the, it doesn't have the highest toxic dose level, which means that this isn't exact, don't quote me on this for vancomycin, but in order to give you guys an idea of what this means, let's say the toxic dosage level of vancomycin was 40 grams, but the therapeutic dosage level had to be 30 grams. So the most a person can take before harm starts is 40 grams, but it still takes 30 grams for it to do anything. You see how that is now, let's see, 40 over 30. I cannot do simple math in my brain. It's about 1 1/3. So 1 1/3 is the chemotherapeutic index. I probably could have picked an easier number, but you see how we have 100 versus 1 1/3? The higher the chemotherapeutic index, the more effective and less toxic it is, right? So here, this is another example. So, and then we have that lower one of 1 1/3. it's going to be less effective and it's going to be more toxic, right? This idea right here sometimes trips people up, but it's still a very important idea. Okay. You guys know spectrum of activity, I think, pretty well. You guys can draw this, it helps. But mostly it's these three that are important. Important to write down. Oops. Know the advantages and disadvantages, right? So if you have a broad spectrum, it is pretty harsh, right? It's going to destroy a lot of things, and it's probably going to target something other than the cell wall. The cell wall is one really, really easy one to differentiate bacteria from human cells. So if we start doing something at the metabolic rate, so if we start targeting ribosomes, we have ribosomes. That could be toxic to us too. But narrow spectrum, it helps us because it's going to decrease destruction of normal flora, and it's not going to have as much resistance. That's why if you're in a hospital, you should, if you were a doctor prescribing antibiotics, you should culture if you can. Culture if you can. Because if you culture, find if it's gram positive, gram negative, you can then give it a more targeted drug. But if you're like, this patient is dying of an infection, we need to figure it out quick, you give a broad spectrum. So at least the damage that you're doing is less than the damage that the bacteria is doing. It's kind of like a catch-22. It's really difficult, but broad spectrum is kind of like one of those things that it's like emergency use and when you don't know what it is. But if you have time, culture it. Culture if you can. Let's see. Yes. So super infections. So let's say you're taking a really harsh antibiotic and it is a broad spectrum and it knocks out your normal flora. So now your normal flora is knocked out. We can then have another opportunistic pathogen come in and recolonize. This is scary stuff. When new bacteria comes in and recolonizes, we now have two separate infections going on by potentially two different bacteria. These are super infections, and it's really hard to treat them, and they're not usually susceptible to any microbials. Because remember, we're giving an antibiotic and they're getting an infection. Like that's scary. Not only that, but you also have to think, their normal flora is gone, which means they have no line of defense in that term because they compete. Normal flora competes for nutrients, space, oxygen. So let's say one E. coli lands on your arm. The reason why you don't get a full-on colonization of your arm, even though there's plenty of stuff for E. coli to latch onto, even though it doesn't colonize your arm, it still existed there at one point. So what happened was this one E. coli, new kid on the block, came in, strolled in, and your staph, epidermidis, your other, skin microflora said, "Who are you?" and kicked it out and basically killed it. Like they were so hardcore that they said, "You're not getting food. We're going to literally suffocate you and not give you any space, and you're just gonna die, and we're gonna live on happily." Like it's brutal. So that's kind of the idea of, bacteria are competing with each other. Now, when we say a bacteria finds a way in the lab, it's because if they are given, like, it's kind of like if you're given somebody a straw bed, right? It's not like their normal bed at home, but they'll make do. So if we give E. coli 3 different sugars and it ferments one, it might start fermenting the other one if it runs out, because bacteria will find a way. It'll start, it doesn't like to ferment this sugar, but it's going to, because that's how it survives. But if one E. coli lands on your arm, it literally can't. There is no way, because there's no nutrients, no space, no oxygen. Okay. This is just an example. Kind of just know how this example interacts with each other, and yeah, I don't think that there's going to be a question on the exam specific to that example. Know these two definitely. Let's see. Modes of action. Now this is one of the Quizlets that I made. I also made a Quizlet more in tune to the actual lab, and a couple other kind of things that I did. Let's see. Alrighty, so penicillins, bacitracins, cephalosporins, and vancomycin. They all target the cell wall. The cell wall causes cell lysis. So if you get stabbed and your insides spill out, are you likely to live? Probably not. Thanks to modern medicine, maybe, but if you were left to your devices in the wilderness, like bacteria is left on your skin, if you are going to have your cell wall sliced open, you're probably not going to live. So these are cell walls. Antibiotics that target the cell wall are most effective against gram-positive, effective against some gram-negative, because remember, they have a cell wall, but they're not effective against fungi and archaea because they do not have cell walls. Okay, that's selective toxicity. The whole idea is I know it's cut off here, but the beta-lactam ring. You guys learned about it a little already, but it cuts this peptide bond between the NAG-NAM, and this is the beta-lactam ring. It cuts this link right here and renders the antibiotic useless, absolutely useless. It is no longer effective. And that's the scary thing. It can just be like snip, and then all of a sudden, what is it going to do? This is just a picture of cell lysis. Now, these are different. The whole idea is that these are different classes of antibiotics that all have the beta-lactam. So penicillin and methicillin are both penicillins, but carbapenem is empamenum, and cephalosporins is cephalothin. And they all have this beta-lactam ring, which targets the cell wall synthesis, the NAG-NAM. See? All right. You don't need to know penicillin G versus penicillin V. Do know that it's usually parentally. Because we have to think about if it's ingested, will it last through the stomach acid? And then also know what prophylactic means. Okay. I'm pretty sure she went through that in lecture. It just means kind of just beforehand. If you're going to go into surgery, they give you antibiotics prophylactically to prevent any bacteria that may live on your skin or may live in a certain area to possibly take advantage of an open wound or part of surgery. Okay. Yeah, these are all just derivatives. Cephalosporins also not the first drug of choice, but think about the allergies. My sister personally has a penicillin allergy. She is kind of screwed, but the thing is, we can try to give her a cephalosporin, but 15% of people who are allergic to penicillin are also allergic to cephalosporins. So I hope she never gets a really bad infection, because that would be no bueno. Let's see. Yeah, cephalosporins. Think about this 1/4. Vancomycin. Now I was talking about this being a really tough drug. Look at how big it is. We were just looking at all these cephalosporins. Look at this thing. This thing is a monster. Jeez. But this means that it's not able to go through gram-negative bacteria. Because of how big it is. It's not going to be able to pass through those pores. Usually it's used to treat MRSA. Okay. MRSA is a tough beast. Remember how it can potentially live on the skin of RNs? That's crazy, right? Last resort drug. Yeesh. All right. I believe that's it for the oh, I found his ads. Let's see. This one. Oh, this is for Mycobacterium. But this one targets the atypical cell wall. So it's not the normal cell wall of gram negative versus gram positive. This is the waxy mycolic, the mycolic acid. So yes. Let's see. So here, we can in bacterial cell walls, they're pretty easy to find, loopholes in order to target them and not us. But fungal walls, we have to get a little more creative because they are composed of polysaccharides, but they're just not the ones found in mammals. So they're not the ones found in our cells. That's one of the few differences that we have. You don't really need to know this. I'm going to be completely honest. But that's the inhibition of cell wall synthesis. You should probably go through, outline these, make flashcards on your own. She will probably point out, or if you want to ask me what would be important, what's not important, I can do that. Or if you guys want to tell me if you think it would be beneficial for me to walk you guys through this, let me know. But I'm going to stop the recording here for today, and I hope you guys have a great rest of your night, and I will see you tomorrow morning at 11:30. Okay? Good night. Bye.